Thank you to the organizers for the opportunity to present this work. I'm presenting this on behalf of the University College Cork and our collaborators at Mead Johnson Pediatric Nutrition who funded the study. I'm going to be discussing the Lactobacillus rhamnosus GG soluble mediators and the effects that we saw in an animal model of early life adversity, particularly maternally separated rats that were fed this treatment, and then we looked at visceral pain, hypersensitivity, and associated changes in spinal cord gene expression. So like many of the speakers these past couple of days, we're interested in the bidirectional communication that exists between the brain and the gut. We know that stress that occurs during developmentally critical windows has long-term effects on the host in terms of behavior and physiology. This is true, obviously, in humans, but also true in our animal models. For those of you that were at Professor Krein's keynote lecture on Thursday, he described very clearly, I think, the effects that we observe in a rat model of maternal separation and how we use that as a model of early life adversity. And the effects that we see in these animals has been really well described. It has been shown to result in stress hyperresponsivity when we look at the plasma corticosterone, changes in behavior like depressive-like behavior, anxiety-like behaviors, visceral pain hypersensitivity, and of course associated changes in reductions in the diversity of the intestinal microbiota. Now, our previous work that we did in collaboration with Mead Johnson Pediatric Nutrition, we looked at the probiotic Lactobacillus rhamnosus GG, so LGG, and we found that it had potential beneficial effects in this rat model of early life stress, maternal separation. So the further study that we've conducted, and then I'm presenting the results from here, we're looking at soluble mediators from the LGG, so referred to in this talk as LSM, so LGG soluble mediators, and we administered them to rat pups that had been subjected to early life maternal separation, and then were fed this treatment from weaning to termination. Excuse me. So this is the experimental timeline that we used. We had Sprague-Dawley rats that were either randomly divided into non-separated or maternally separated groups. For those that received maternal separation, they were separated from their mothers into a separate housing room every single morning from postnatal day 2 to 12 for 3 hours, and then returned to the home cage with the mom. And then they were, after postnatal day 12 to 21, they were just left in a normal situation with the mother. Weaned at postnatal day 21, and at that point randomly allocated into treatment groups. So our controls received drinking water and standard rodent chow, and our LSM group received the LGG supernatant and via the drinking water and standard chow for food. At postnatal day 79, so roughly 11 weeks of age, we subjected them to colorectal distension as a measure of visceral pain hypersensitivity. Then approximately a week later, these rats were exposed to a half-an-hour restraint stress, and then we took tail bleed from them to assess plasma corticosterone. And then at postnatal day 85, they were killed, and we took the spinal cords, brains, blood, we took fecal samples throughout. As I said, they're Sprague-Dawley rats. We had an N of 12 per group. They were all males that we used throughout the experiment from weaning onwards. And So just briefly about the LSM preparation, I can say that it's an LGG supernatant. It was collected during the late exponential growth phase of the bacteria. It was subjected to desalting methods, ultrafiltered and lyophilized, and the rats were given a dose equivalent of 1.3 to 4.3 times 10 to the 8 CFUs of viable LGG per animal per day, or the equivalent of. So the behavioral test that I'm showing here is the colorectal distension that I'm sure most of you are familiar with, but you insert the balloon into, or a deflated balloon into the rectum of the animal, and then it's inflated to various pressures, and the rats are assessed for pain responses. And this is a very well-established model of visceral pain hypersensitivity or sensitivity. So for my results here, so the non-separated animals are on the left, the maternally separated on the right. The black bars are the animals that were in the control treatment groups, and the blue are those that received the LGG soluble mediator in the water. As you can see, and as we've replicated many times in our group, the maternal separation process, or the maternal separation experience, I suppose, increased visceral pain sensitivity. So at a reduced balloon pressure, they demonstrated pain responses due to the increase in balloon pressure. However, the LSM-treated animals showed an amelioration of this pain hypersensitivity. So they didn't show pain responses until it was reaching higher balloon pressures. For our measures of stress responsivity, again, it's the same setup as before. So the maternally separated controls show an increase in plasma corticosterone response after a half an hour restraint stress compared to the controls, and this was reduced in the LSM-treated animals. So the plasma corticosterone returned to similar levels to that of the non-separated control. We did a whole, we took the spinal cord to the lumbosacral region of the spinal cord and sent it for whole genome sequencing, and I'll just direct you to the Venn diagram here. So the green refers to an upregulation of about 63 genes that we saw if you compare the non-separated controls to the maternally separated controls. So maternal separation resulted in an upregulation of 63 genes. And then the red circle refers to, there were 199 genes that were downregulated if you compared the maternally separated LSM treated to the maternally separated controls. But we were particularly interested, of course, in these 41 genes that are in both of these circles because it showed that the genes that were upregulated by maternal separation were, in fact, downregulated after the LSM treatment. So when we particularly focused on these 41 genes, we did a David analysis, and we see that they genes have been reported to be involved in various cellular processes, but of particular interest to us, there was the response to steroid hormone and response to corticosteroid that we were particularly interested in. And again, also 12 of the 41 genes that we looked at, or that were changed by both treatments, have been reported to be associated with either stress, pain, or both. We were particularly interested to see annexin 1 pop up because the annexin receptor, the annexin 1 receptor, has been shown to be responsive to bacterial products. We looked at the microbiome of the fecal pellets throughout the course of the study. This is at the end of the study. We have our 4 treatment groups. Oh, what have we done? 4 treatment groups in concentric circles represented here. At the family level, we saw that the LGG soluble mediator increased the levels of rhamnococci compared to controls. And at the genus level, which is what we have shown here in the figure, we see that the maternal separation increased Tannerella if you compare this to the non-separated controls, and the LGG soluble mediator prevented this increase, and that's represented by this asterisk here. Additionally, maternal separation decreased the Clostridium compared to the non-separated controls, and again, this was normalized after the LSM treatment. And then we did we looked at bacterial diversity, and we see that, as again we've shown many times in our group, that maternal separation resulted in a decrease in diversity, but this was increased after the animals were fed the LGG soluble mediator. So to summarize, administering the LSM, or the LGG soluble mediator, to rats that had been subjected to early life stress, these animals showed an amelioration of the visceral pain hypersensitivity in colorectal distension. They showed an improved corticosterone response to an acute 30-minute restraint stress. There were gene expression changes in the spinal cord that we could associate with some of the pain responsivity that we observed. And the LSM also counteracted the reduced diversity that we observed as a result of maternal separation in the fecal microbiome. So these findings suggest a potential role for nutritional interventions, particularly with probiotic soluble mediators, and in particular if you're looking at pain responsivity. And our current work now is further identifying mechanisms of action. So I would like to thank my co-workers at the University College Cork, particularly Professors Cryan and Dinan, as well as Siobhan O'Mahony, Catherine Stanton, and Matteo Bischetto, who was a postdoctoral fellow also heavily involved with the study, and then our collaborators at Mead Johnson Pediatric Nutrition. So thank you very much.
Thank you. Paper is now open for discussion. Maybe I'll start out. Any idea what those soluble mediators are? Because obviously the question is this specific to the bacteria, and this is something that does not really need to be from the bacteria, and what's your take?
Sure. Yeah, we often get asked that question. What I can tell you is that Mead Johnson has obviously been looking at this, and in vitro studies they have found that the soluble mediator has immunomodulatory effects, and that if they apply proteases, those seem to disappear. So they're pretty sure it's a proteinaceous mediator. Having said that, in chromatography work, they're also seeing that it could be a polysaccharide. So, yeah. Yeah.
Any more questions from the floor? Please.
It was an obvious question to
Yeah.
pose, but if you saw the results, you also saw that something happened in your control animals if you administered your extracts.
Yes.
And it was often not in the same direction of action. Do you have any comment on that?
Yeah, we often do see actually that the both the LGG and the soluble mediators had effects in the non-separated groups. Of course, they're not usually as large because you're maybe perhaps dealing with the ceiling effect if you don't have a challenged group. In terms of the opposite direction, hard to say, I suppose. Maybe it's the double exposure to early life stress changes responsivity, I would say. Yeah.
Or the kind of proteins you're balancing?
True.
Sir?
From your work, can you say that the effect continues after you stop giving it, or you can't answer that question? In other words, does the soluble media have to be present on the day that you do these measurements to see the effect?
Yeah, we haven't done those experiments in this study. These animals were fed from weaning up to killing, so they've received daily doses. It often seems in probiotic type studies that you do have to do a continued treatment in order to see effects. But I couldn't answer that from this particular study.
Do you have any idea on the stability of these mediators? For example, are they sensitive to acid? I'm referring to the way of the route of administration if you use this in a human, obviously.
Yeah, if you use this in a human. Well, this was delivered via drinking water, and in terms of sensitivity to acid, I'm not sure actually. I couldn't say really. I haven't conducted those experiments.
And the dose you used, would there be a dose that would be comparable to what you would use in a human?
They're comparable to. It's comparable to the dose that you use in the probiotic study. So it's considered comparable to a human dose of probiotic. Yeah.
There are no more questions, then I'd like to close this session. Thank you
Thank
very
you.
much.